Hello, guys, welcome to the Shivas of the Medical Cannabis Chronicles. Today, we're gonna talk about the roots of cannabis, episode 1, where it all began. So, we have an expert today. Let's start with something I keep hearing that cannabis is somehow related to beer. Is that actually true?
It is. Cannabis belongs to a plant family called Cannabaceae, and its closest living relative is Humulus hops, the same plant used to flavor beer. So they are genuinely cousins. They split from a shared ancestor around 28 million years ago.
28 million, not 1000, right?
Million. That's the part people find hard to picture. The plant has existed in the wild for tens of millions of years. Humans only enter the story much later.
So, where did it originally come from?
The best evidence point to the northeastern Tibetan Plateau near Qinghai Lake, though getting to that answer took some clever detective work.
Why detective work?
Because cannabis and hops pollen look almost identical in the fossil record, you can't easily tell them apart. So researchers read the neighbors instead. Cannabis pollen turn up in open, cold grassland alongside plants like sagebrush. Hops turns up in the forest, match the surrounding environment, and you can work out which plant you are actually looking at.
And for all those millions of years, it was just growing wild?
Exactly. A tough, vine pollinated weed of the cold open country, slowly drifting across continents. No humans, no farming, just a survivor on a high plateau.
So, when did humans actually start growing it then?
Only about 12,000 years ago. Put that next to the 28 million, and it's striking. The plant was wild for tens of millions of years, and we have been evolved for barely 12,000. In its story, we are a footnote.
Oh, and where did that domestication happen?
East Asia, a single origin from an ancestral gene pool that survives today in wild and traditional plants in China. A large 2021 genome study basically rewrote the textbook on this. People used to assume Central Asia. The DNA points to East Asia.
And was it grown for its compounds from the very start?
No, and this is the elegant part. Early on, it was a multi purpose crop fiber, seed, and its compounds all at once. The split into fiber type hemp and drug type plants came later, only about 4,000 years ago. So, divide we treat as fundamental today is, in elementary terms, quite young.
So, does that original wild plant still exist today?
Probably not. The truly wild ancestor is most likely extinct. The closest we have left are those feral descendants in China, which is exactly why they are so scientifically valuable.
Let's go into the genetics now. How does the plant actually make cannabinoids?
The key thing is that cannabinoids are not each made by their own separate machine. They come off one shared assembly line. Two streams of raw material combine into one central molecule called CBGA. People call it the mother cannabinoid because nearly everything else is built from it.
So, what makes the different cannabinoid different one than the other?
A small set of very closely related enzymes, they each take the same CBGA molecule and turn it into a different product, THCA, CBDA, or CBCA. And those enzymes are not separate inventions. They are duplicated copies of a single ancestral gene that drifted apart over time, sitting at essentially one tightly linked spot in the genome.
Oh, that's cool. How does that connect back to the hemp versus drug split?
beautifully. Actually, because those enzymes compete for same raw material, breeding a plant hard in one direction often breaks into the other gene. Select for high THC and the CBD root shuts down. Select for fiber hemp and the THC making gene switches off. So the difference between a drug type plant and hemp comes down largely to whether one gene, THCA synthase, is working or broken. That's one of the main genetic markers used to tell them apart.
That's very interesting to know. Does the living plant actually contain THC and CBD from this very starting?
This is the bit that trips people up. No, not directly. The plant makes the acid forms. It's his that converts them into the familiar active forms through a process called decarboxylation, which is why raw material and a heater or processed product are chemically different things.
So, if you had to sum up the entire whole story in a few sentences, what would it be like?
A cold climate plant splits from hops around 28 million years ago on the Tibetan Plateau. It spends millions of years as a wild weed spreading across continents. Then only 12,000 years ago, human in East Asia domesticated first as a multi-purpose crop and later split into fiber and drug types, and the tool behind that split is a small family of duplicated genes funneling one shared molecule into different products.
Anything people commonly get wrong or should be cautious about?
Two notes, two honest notes. First, those deep type dates come from molecular estimates and fossil pollen. They are the best available, but they carry real uncertainty. And the second, the popular Sativa versus Indica labels don't map cleanly onto the actual genetics. That mismatch is a whole topic in itself.
Okay, which is where we're going next in the episode.
Exactly. That's episode 2. We will get into what sativa and indica really mean and what the genome actually says.
Perfect place to stop. Thanks for walking through us, and thank you, listener, for listening until here. Keep tuning until next episode, and please do subscribe and comment if you have any opinion or if that's any information that we need to take. Thank you so much.
Thank you guys.